Hello, welcome to the genetics lecture for the PG3 course 2025. Psychiatric genetics is likely to have a major impact on clinical practice in the next 5 to 10 years. It's having a distinct effect in the area of autism spectrum disorders, as we'll see in a later lecture. Here we're going to go through the introductory concepts. And here they are. I will be going through each of these one by one. The human genome has has over 6 million base pairs. About 20,000 genes code messenger RNA for a protein. About another 25,000 code RNA, but these don't result in a protein. We used to refer to all of the rest of the DNA as junk DNA. That really is an obsolete term. Much of this DNA now is regulatory or structural and thus does in fact play a significant role. Here we can see the regulatory elements. Here's the transcription site. The polymerase will transcribe the DNA into RNA, and all of these different transcription factors will either enhance or suppress transcription. And these often are influenced by other areas of the DNA. An enhancer area is activated when an activator attaches to it. It will actually bend the DNA over and influence the transcription factors. And similarly, if a repressor protein binds to the silencer sequence, that too will limit transcription. So enhancer and silencer refer to the DNA sequence, whereas activator and repressor refer to the proteins that bind to it. And of course, those proteins are transcribed by other sites, thus the regulatory DNA having a major impact on ultimate transcription and protein production. Now, our first step in looking at genetics is to examine the familial aggregation by calculating the relative risk. So first we look at the risk in the general population, which is 0.8% for schizophrenia. Depending if you have what kind of relative you have who has schizophrenia, your own risk will be increased. If we combine the risk in all the first degree relatives, that comes to about 7%. Now we divide 7 by 0.8 and we get the relative risk, or lambda, which is 8.75. Any rate above 1 indicates a elevated familial risk. Now the first concept we're going to look at is heritability, and this is key to understanding many aspects of behavioral genetics. So the older way of finding heritability is through twin studies, and also this makes it easier to understand. So let's imagine we have two groups of twins, one monozygotic and the other dizygotic. If we correlate the heights of the monozygotic twin pairs, it comes out nearly perfect, 0.99, because nearly every twin is the same height as his co-twin. In contrast, the DZ twins only share 50% of their genes, so while their heights will be similar, the correlation will not be as strong. And as you can see here, it is 0.54. So the fact that the MZ twin correlation is significantly higher than the DZ twins tells us that genetics are implicated in this trait. Now we can also do this with a categorical measure. So we assess the twins for the presence or absence of autism. You can see in the MZ twins, if one twin has autism, then there's a 72% chance that the other will have autism. Whereas with the DZ twins, if one has autism, there's only a 3% chance that the co-twin will have autism. So that again clearly implicates genetics in autism. Now, it's important to bear in mind that heritability is a measure of how much of the variance of a trait in the population can be attributed to genetics. So if we have three individuals, then the difference in those three individuals is what we're interested in as far as heritability is concerned. Heritability says nothing about the mean of the population. So let's take an example of a village that is in an underdeveloped part of the world. Nutrition is very poor, has been for generations, so the people are significantly shorter than people in the developed world. And here, though, we will have a bell curve with most people in the middle, a few relatively tall people, and a few relatively short people. So nothing changes. This first generation has children. We will find that the mean of the population is the same. But relatively tall parents, C1, will have relatively tall children, C2, and same thing with B1, who are shorter. Their children, B2, will also be among the shortest in the village. And this is because heritability is high, this position within the population is heavily influenced by genetics. Now, let's say we then introduce agricultural improvements so that generation 3 grows up in a much more healthy and nutritious environment. What will happen then is generation 3 will be taller, the entire population will shift to the right, but the rank ordering of the generations will really not change that much. The relatively taller people in generation 2 will have the relatively taller children in generation 3, and so on. The difference between the individual is still determined by genetics. Now, we can calculate the heritability simply by subtracting the DZ correlation from the MZ correlation, multiplying by 2, and we get the common finding that the heritability height is about 0.9, meaning 90% of the variance is due to genetics. So if A and B differ by 10 inches, then 90% of that 10-inch difference, or that 9-inch difference, can be attributed to genetics, or that variance. Now, we can do this with a whole variety of traits. And then when heritability is less than 1, the remainder of the variance is accounted for by the environment. And we divide that into shared and non-shared variance. The shared variance is events that the twins have in common. So they have the same socioeconomic status, the same parents. Non-shared can be things like where the twins are different. One can have a favored position in utero. One twin can have a head injury and the other does not. One can go into combat and the other did not. That was the basis of a major study in the Vietnam War that we'll look at. Or one twin can have a very positive experience in school and the other does not. So if we look at IQ, or intelligence, about half the variance can be attributed to genetics, and the rest is with the environment, and the majority of that environmental influence, not surprisingly, is the shared environment that the twins have. Now, we look at extroversion, the personality trait, that also has a relatively high heritability, 0.6, but most of the environmental variance is non-shared. So maybe one twin got put in a school play, and that contributed to him being an extrovert. Now look at bipolar 1. Its heritability is almost as high as height. Almost all of the variance is attributable to genetics, very little to environment. With major depression, the heritability is about similar to that of IQ, but ordinary anxiety, neurotic anxiety, has virtually no genetic contribution to the variance at all. Now that may not be true for OCD or severe panic disorder, but when you talk about generalized neurotic anxiety, that is the case. Schizophrenia has a heritability about 0.6, similar to type 2 diabetes. Very little genetic influence on the variance for bulimia. And interestingly, look at same-sex attraction. Surprisingly low heritability for a trait that is generally very fixed in individuals. So what accounts for the rest of the variance? It obviously must be environmental, and the in utero environment may play a role. If we have time again, we'll look at that in later lectures. The human behavior is virtually never explained by a single gene. We have multiple genes working together to kind of create the classic bell curve. And when multiple genes act in an additive fashion, it's very simple. So if there are three risk genes that increase your risk for the disease by 5, 3, or 6%, if you're unlucky enough to get all three, your risk is now 14%. So the and this is part of calculating a polygenic risk score that we'll talk about in a moment. Now, occasionally there are non-additive effects. Genes combine in weird ways to either dramatically increase or dramatically lower the risk. This drives geneticists nuts, and maybe part of the reason we have a hard time working out all the genetic effects for various diseases. Now, I want you to have these terms down cold. So on the upper left, we see that the trait is polygenic. Many genes contribute to the trait. Now to the right, the gene is pleiotropic. One gene is contributing to many traits. So traits are polygenic, genes can be pleiotropic. Now, two related concepts are first variable expressivity. Notice that the same trait is there, but it occurs in the different individuals to a different degree. It's a quantitative difference. Perhaps one of the best examples of this is male pattern baldness. If we look at the royal family of Great Britain, King Charles and his two sons, Harry and William, all have very different levels of balding, and that's typical among any group of male siblings. That's variable expressivity. Incomplete penetrance, in contrast, refers to whether the disease is present or absent. So 4 individuals get the gene, but only 2 people get the disease. So the disease has 50% penetrance. So again, penetrance refers to the disease being either it's there or it's not. Variable expressivity refers to that there are gradations or quantitative variation of the trait. Now here, with just 3 genes, each having a dominant and recessive allele, we can very easily form a bell curve. So that shows how genes working together can create normal variation. Of course, we have thousands of genes in most human traits. And again, psychiatric disorders are both polygenic and pleiotropic. So on the left, this is a schematic. You see each of the 3 genes contributes to different disorders, and each of the disorders, so the genes are pleiotropic, and each of the disorders gets input from many genes, and thus the condition is polygenic. On the right, you see a more literal example. What they're illustrating here is that there are a set of genes, the general genes, that contribute to all psychiatric disorders, whereas there are separate sets of genes, the externalizing genes, that contribute to all of the various acting out disorders like ADHD, drug abuse, and antisocial personality. And then there are another set of internalizing genes that tend to contribute to all the depressive and anxiety disorders. Now, the next concept I want to look at is gene by environment interaction. So here we have a gene that has 2 variants. In people who inherit variant 1 then are exposed to 2 different environments. If they're in environment 1, they're healthy. If they're in environment 2, they get a minor version of the disease. In variant 2, if they're exposed to environment 1, no disease. If they're in environment 2, they get major disease. So environment 2 contributes to the cause of the disease, but the people with variant 2 are much more vulnerable. That is the gene by environment interaction. Now I'm going to give you a specific example involving a gene for the enzyme monoamine oxidase. In the brain, this breaks down norepinephrine and serotonin, and genetically it comes in both a low and high activity subtype. And there's an interesting relationship between these subtypes and criminal activity and exposure to child abuse. So we see on the x-axis the increasing severity of childhood maltreatment, and you can see for both lines, the more maltreatment, the worse the antisocial behavior. But the people with the low MAO activity are particularly vulnerable, particularly at very high levels of child abuse. They are much more likely to develop antisocial behavior than those with the high activity. This finding has not been followed up, I think, as strongly as it should, but I present it here as an example of gene by environment interaction. All right, now we're going to get into molecular genetics itself. So allele is just the variant forms of a gene at a gene location. It can just be a normal variant or can have functional significance. When we say it's associated, we mean that those people who have the disease have a certain allele that travels with that disease or is associated with that disease. And then finally, linkage means we know that it moves from one generation to the next. Now, the first type of variant we're going to look at is the single nucleotide polymorphism, SNP, which is commonly referred to as a SNP. So enzymes will chop the DNA into different pieces depending on where the SNP is. So here the SNP is a G, here it's a C. So the enzyme lets this remain as one long DNA strand, and this is split in two. So on the gel, this person is different because the two fragments go further because they're smaller. Repeat that thousands and thousands of times, then you can create the DNA fingerprint for each person. In this case, this depends, which allele you get depends on whether you more easily can taste bitter things or not. So here is a good example of how a SNP is occurred. The CG gets replaced by the TA or vice versa. It's sequent variation. There's no change to the overall structure of the DNA. We generally discover these by a genome-wide association, whole exome sequencing, or whole genome sequencing. And if our markers are near a gene, then obviously these two people, their gels will look different. And if people with both SNP1 and SNP2 tend to have a high prevalence of a disease, we say, aha, maybe the gene that's near those SNPs is related to that disease. So here's an example of a simple association study. We have a hypothesis that type 1 diabetes is an autoimmune phenomena. So we look at the type 1 diabetes patients as to whether they have a certain variant in the HLA system, and indeed they do. The DR4 is more common. It's statistically significant, so we say that HLA DR4 is associated with diabetes. Now, classically in an association studies, we develop a candidate gene. So naturally, early in the game, we were looking at dopamine genes in schizophrenia and serotonin genes in affective disorder. Now, an association study can be very sensitive, but it's very prone to error and bias. And you get, if you're just doing a candidate gene study, picking a gene of interest and testing it, you are very likely to get a false positive. So a way around that is to do a linkage study. So you look at the marker and then you look to see if that marker associates with the disease in one generation and then follows the subsequent generations. Now the obviously problem with this study is you need large families and you need access to 2 or 3 different generations. That's methodologically very difficult, so very few of them have been done. And the ones that were done really didn't yield much interesting data. So while the linkage study is talked about in textbooks and in board exams, it's really become mostly obsolete in psychiatry today. What is the big deal now is the genome-wide association study. Here, we don't have a hypothesis. We don't pick a candidate gene. We look at very large samples and we examine our markers across the uh, entire genome. We obviously do our statistical adjustments, and then we can have more uh, faith in our results. So when you do a GWAS, a genome-wide association study, you generally present the results as a Manhattan plot, so named because these things look like skyscrapers. So on the x-axis we have all the different chromosomes. Each little dot represents one of these SNPs that are associated with the disease, or could be associated with the disease. And the red line indicates where they are statistically significant. So we can see here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 loci, 12 spots on chromosomes are associated with the disease, and many of these have hundreds of SNPs that could be contributing. Now, increasingly, the GWAS is replacing the twin study. We got all these SNPs. We add up the risk that each SNP conveys, and we get an SNP heritability estimate. Now, when we do that, we find an odd thing almost all the time, that the SNP estimate is lower than the twin study, and we refer to the difference between the two as the missing heritability. What does that mean? Well, stay tuned. Before we go to the missing heritability, though, let me talk about what we do know. If we add up the risk from each SNP, we get a PRS, or polygenic risk score. Sometimes it's called a PGS. And the higher your PSR, the higher your risk for the disease. And then we can correlate the PSRs for different disorders to determine if they share genetic risk. So what we see here is, obviously, if we find all the SNPs that are associated with ADHD, number of those will correlate with the rating of ADHD symptoms. If we correlate the PRSs of obesity, that's going to correlate with weight. But now what we find is that the PRS score for ADHD correlates with the PRS for obesity, meaning that the genes are pleiotropic. Genes that put you at risk for ADHD also put you at risk for obesity, and vice versa. And here's some interesting examples of how the polygenic risk score can be used. So here, the correlation, blue is positive, red is negative. So having the PRS for ADHD correlates with using a mobile phone more often, having used it in the last 3 weeks, and did you use a hands-free device when you're driving? No, you do not. That negatively correlates with genetic risk for ADHD. Do you play video games? That positively correlates with the genetic risk for ADHD. Now let's look over here at schizophrenia. The genetic risk for schizophrenia is negatively correlated with mobile phone use and with computer games. But for autism spectrum disorder, PRS correlates negatively with phone use. Autism people are less social, but in fact, in contrast to schizophrenia, people with autism spectrum disorder do tend to be obsessive about video games. So this shows us we can correlate the genetic risk with a wide range of traits. Now, here's some examples of how we've used these SNP studies in things beyond psychiatric disorder per se. So there's a trait called risk tolerance. You know, how risk-taking are you? And not surprisingly, people who are risky will drink more, have more sexual partners, are more likely to have had speeding tickets, etc. So in this study, they looked at the 23andMe data with over 1 million subjects. We know from twin studies, risk tolerance heritability is about 0.3. And what they find is that the SNP heritability is about 0.156, And I believe you can make sure I show you where this is here. You just right there, there they are added all up on the bottom row. So you subtract 0.156 from 0.3, the 0.144 is the missing heritability. What's interesting about this is that those genes tend to be dopamine and serotonin genes. Well, if you were listening in the last lecture, you probably are not, hopefully, are not too surprised by that, because I talked about dopamine being the go neurotransmitter and how serotonin is involved in impulse control and mood. So the fact that these genes would influence risk tolerance makes sense and tends to validate the study. Now, how about IQ? As I told you earlier, the twin studies show heritability about 0.5. So in this study in Nature, they looked at IQ in 16 different data sets. What did they find? Well, they found 205 loci that involved over 1,000 genes. So anyone who's going to do genetic engineering for intelligence, you're kind of shit out of luck, because there's just too many genes to mess around with. Most of these genes, not surprisingly, are expressed in the brain, are involved in synaptic structure. And you can see that here. All of the pink ones on the left are the brain. That pituitary sticks out there, but that is not statistically significant. So yes, intelligence is the brain, in the brain. We proved it. Now, still though, the SNP heritability is only 25%. Where's the rest? Well, some of it could be this interactive effect non-additive effect that I talked about earlier, but there's another piece of it that's coming. Now here, this is very interesting. What they've looked at is the PRS for IQ in people of different socioeconomic groups. Now, you might argue, well, people at the top of society, that's the cream of society. They must be genetically superior because they're so brilliant. They've risen to the top of society. You could probably think of at least a couple people currently in the news who think that. But in any case, when they actually looked at it, they found the opposite. That among poor people, they had more of the PRSs for IQ than did the rich people. Well, how would that be? Well, if you're a dumb rich kid, you have an enriched environment that kind of helps you along. You don't need a big PRS for IQ. So it might be the opposite of what sort of genetically obsessed people might think. Now, missing heritability, where is it? Well, one place is in this next section, the copy number variant, CNV, which is structural variation. So you see there that an area of the DNA has been duplicated. The structure has been changed. And we usually detect these by chromosomal microarray, or CMAs, although increasingly we can use whole exome sequencing to find these as well. So what can happen here is we see these can be either de novo or inherited. So on the left, we have the parents, no CNVs in the parent. And then one of the children has a disease, and we can see that a little piece of DNA has been chopped away. Now on the right, we have a person who's an asymptomatic carrier, and they've got a piece chopped off, and they pass that on to their parent, and now they have a disease. This is an example of incomplete penetrance, because the parent doesn't have the disease, because even though they have the gene, and the child does. As a general rule, the larger the deletion or duplication, the greater the effect of the CNV and the more likely it is to cause disease. All right, so here's just another example. De novo deletion on the left and inherited duplication on the right. Now, this is very important to wrap your mind around. So on the x-axis, we have how frequent the genetic mutation or variation is. So SNPs are very, very common. So they're at the far right. Now, on the y-axis, we have the effect size. How likely is it that the genetic variation will cause the disease? Well, you can see for SNPs, it's very low. each SNP only contributes a tiny amount of the risk. If the CNV is very small, it also, in addition to being very rare, only contributes a tiny amount of risk. On the other hand, if we get very large CNVs, large duplications, or large deletions, these can have a large effect size that sometimes can approach single gene effects. One example of this is DiGeorge syndrome, 22q minus deletion syndrome. We'll be studying that disorder in a later lecture. So SNPs are always very common and very low effect. CNVs are always rare, but their effect depends on how big they are. And in journals, this is how it is presented. This happens to be a study of ADHD. We'll look at this later in more detail. Here are all the SNPs in these different genes. Very common, but very small effect. Here are a bunch of medium-sized deletions and duplications. These do have a stronger effect for ADHD, but they're rare. And this is extremely rare, but very potent. This MAP1, MAP1A deletion. And this is pleiotropic. It has an effect for both ADHD and autism. But it's very rare. You can see hundreds of thousands of people, genotype them all, and there'll only be 2 or 3 patients that have the variation in MAP1A. But if they have MAP1A, they surely have ADHD and or autism. Now, these CNVs are responsible for a number of neuropsychiatric disorders. Here's DiGeorge syndrome, velocardiofacial. Here's the famous deletion at 15q11.13. If the deletion is in the maternal chromosome, you get Angelman syndrome, the happy puppet syndrome. If the deletion is in the paternal chromosome, you get Prader-Willi, the hyperphagia, and so on. And these disorders, some of them are not named yet. They may or may not be associated with autism or schizophrenia. More about that to come. The main thing for you to understand right now is that the CNVs, their effect is based on the size of the deletion or the duplication, and they tend to be particularly important in neurodevelopmental, developmental delay disorders. Now let's talk about epigenetics. Epigenetics means above the genome, and ultimately epigenetics is important in every cell in your body. Every cell has exactly the same DNA sequence. Whether it's a neuron or a red blood cell depends on which genes are turned on or off. And the turning on and off of genes occurs via epigenetics. So we're going to be looking at 2 major mechanisms. First, environmental events can cause the methylation of genes, and that tends to silence them because you put a methyl group on the cytosine. On the other hand, we can put acyl groups on the histone tails here, and when the acyl groups are on there, it will open up the histone octamer so the messenger RNA can be translated into proteins and so on. So here's just another example, a more detailed one. Here's the chromatin. We have 2 enzymes, HAT and HDAC, that either put the acyl group on or take it off. We unwind and we get transcription. Here the gene has been silenced by methylation, and here we're reading out microRNAs that do not produce a protein but jump in and help with the regulatory process. This is described in the book.
So that's your box one. So now what regulates gene expression in eukaryotes? Like, how do we know when to turn on or turn off our genes? So far, we've learned about transcription factors being required. If we don't have transcription factors, then we can't do gene expression. But there's more. Transcription is a really common area and like an easy area to kind of control the amount of protein that's being made. but there's actually a whole lot of different steps involved in ultimately regulating the final protein product. We're going to focus mostly at the area of chromosomes and transcription, but we'll touch on in class the end parts as well. So here we have some chromatin. Chromatin you can see up here in the nucleus. Oops. So here we have chromatin in our nucleus. Chromatin is made out of DNA wrapped around histone proteins. And so we can either have our DNA be loosely condensed or tightly condensed. So with this here, we have two kinds of chromatin. We have euchromatin and we have heterochromatin. So here euchromatin is, you can tell by looking at it, it's more loosely condensed. And then our heterochromatin is more tightly condensed. And with that, we want to think about, okay, if gene expression means transcribing genes and producing RNA and then ultimately a protein, we need RNA polymerase and transcription factors to be able to attach to the promoter sequence of the DNA. So if I look at euchromatin and heterochromatin, you can see it would be easier for RNA polymerase to attach to a promoter in euchromatin and much more difficult in heterochromatin. So generally, genes within heterochromatin are not expressed because it's so tightly condensed. And so what we can do is we can modify the histone proteins and the DNA to influence whether or not it's euchromatin or heterochromatin. So when you want to write in box 2 the difference between euchromatin and heterochromatin. Alright, so now when we look at one option, here we have what's called histone acetylation. So this little blue circle represents RNA polymerase. And RNA polymerase is going to, in this case, be blocked from transcribing. If this is heterochromatin and it's so tightly condensed, then RNA polymerase cannot transcribe. So what can happen is they can add what we call acetyl groups to the protein, to the histone. When the acetyl groups attach, it causes the chromatin to unwind and loosen. Now, RNA polymerase can attach to a promoter sequence and transcribe. So histone acetylation promotes or encourages gene expression by opening up that chromatin structure. Okay. However, when the acetyl groups come off, that DNA coils back up to heterochromatin, blocking transcription and gene expression. So here's just showing the little acetyl groups attached to the histone protein. So here if I have some chromatin and I add acetyl groups, go ahead and think in your head what it'll look like. Good, good. It's going to be loosely condensed. It's going to open up and encourage transcription. If I take off the acetyl groups, that's right, it'll go back to heterochromatin. That's called deacetylation. So now I like this picture because it shows when you have acetyl groups, gene transcription is turned on. When you remove or deacetylate, that gene transcription turned off, back in heterochromatin. So how are histone acetylation or deacetylation related to transcription? That's your box 3. And now, when we look at another way that we can influence chromatin, that's through DNA methylation. This one is different because we literally are adding chemical markers to the DNA and map the protein. And when we add methyl groups to our DNA, it causes it to become heterochromatin. And so now it's our DNA that is actually being methylated. So this right here is how, like A, T, C, and G, this is C, cytosine. You can see how there's a methyl group added. So it's an actual modification to our nucleotides. And it causes heterochromatin and turning off of gene expression. So
Very good. So that now I'm going to show you and this again is described in the book, but it is a behavioral example of epigenetics. So here's a picture of a mother rat. They're called dams. And when rats have their babies, they arch their backs so that the pups can fit underneath there, and they also do a great deal of licking and grooming of these rats. This process is very important. Rats who get lots of licking and the moms arch their backs, they have a moderate response to stress. These are the open circles. They see that the ACTH and cortisone is only moderately increased when there's a stressor, a loud noise in the cage. In contrast, the rats that did not get licking and grooming tend to have an exaggerated response. Now, if you take the baby rats away from the low licking mother and give them to a high licking mother, then epigenetics leads to those mice to having a moderate stress response, and they apparently avoid being anxious, for lack of a better word. But you must do this within a certain critical period. If you wait too long, it's too late, and the rats are doomed to have this exaggerated stress response. Now, how does this come about? Again, this is described in the book, but when the mother rat licks and grooms, it activates serotonergic input to the hippocampus, works through the cyclic AMP system to turn on protein kinase A. We phosphorylate CBP. That comes in and will activate the transcription of nerve growth factor 1A. When that's produced, it comes back into the nucleus, demethylates this promoter so that now the glucocorticoid receptor can be transcribed. And if you've got lots of these GRs, when the cortisol comes in, it clicks them on. So that will alter the hippocampal output to the hypothalamus, reduce CRH, reduce ACTH, less cortisol, thus a modest stress response. And this occurs in both genders. Now, for the rats who don't get licking, we got nothing. All of this is dead. We get small numbers of GR receptors, so we get an exaggerated stress response. Now, interestingly, female rats who are licked and groomed will grow up to lick and groom their babies. But the female rats who don't get licked and groomed will continue to be non-lickers themselves. So what makes a licker rat? Well, a good rat, a good mommy rat. Well, when there's licking, prolactin activates the TRK system, activating a protein called STAT5. It gets phosphorylated, moves in, triggers the estrogen, the production of the estrogen receptors. When estrogen binds to those receptors, it triggers the production of the oxytocin messenger RNA and the production of oxytocin receptors, and then oxytocin binding to that receptor produces the maternal behaviors in adulthood. And of course, you see right here, when we don't have TRK system activated, none of this, and without sufficient OTR, oxytocin receptors, we do not have the appropriate maternal behaviors. And this occurs in the medial preoptic area that we'll talk about in the next lecture. And that concludes this lecture. Thank you very much.